Hi, everybody, and welcome to the Bridges from Python to R for Bioconductor via Basilisk. This is going to be a hands-on workshop. We have had some very exciting moments with our workshops, but thanks to Alex, as always, this is actually going to work. And I'm going to start my own little workshop for just, you go to workshop.bioconductor.org and look to see if I've got any workshops running. The answer is no. Yeah? Ah. Yeah. Yeah. I hope I'm logged in as the right person. No, see, I'm logged in as Andrew McDavid. That shouldn't have been. How about that? See, I'm doing this on a different computer, so this is a little different for me, but everybody should get their little workshop going. It may take a little while. Beautiful. So I'm logged in. We have a workshop there that needs to be stopped, it looks like. And now I'm going to scroll down and go to the bridges from Python to Bioconductor, and we'll launch it. And that should come up eventually. All right. Well, welcome everyone. I hope you're having a good time. We are going to be talking about how to make Python and R interoperate through a wonderful software system called Basilisk, which is a work of Aaron Lun of Genentech. He's been involved with Bioconductor for a long time. He was the principal moving force, accompanied by a number of others, behind the orchestrating single-cell analysis with Bioconductor, which is a monograph scale collection of R markdown and data and software tools for single-cell analysis, very rich ecosystem for doing that. And he also wrote infrastructure to make books, and that's what the OSCA book is. But he also wrote infrastructure to help manage your Python dependencies, which is often a complicated thing to do. And so what I wrote here is that we'll initialize our RStudio sessions first, and let's see how that went. See, it's still launching. So you may be in that situation, or you may be a lucky person. But that's okay. Eventually it'll come up. And then some basic reflections on Python and R for data science. I thought there were some interesting remarks in the imaging plenary that we had this morning about the desires that various labs who are heavily involved in imaging have for incoming software engineers, where one of the largest topics that was identified by people who were queried was they want Python expertise. So I think it is pretty well recognized that there's a lot of attitude that when you're doing data science, Python is the way to go. And R is maybe lagging behind. And we'll get into that a little bit. Then we'll start working with a couple of packages that I worked on, scvi-R, to help make a bridge from R and Bioconductor over to the scvirs, particularly scvi-tools, where variational inference is a major concern of the Nir Yosef group out in California. So we'll look at that, and we'll also look at BiocHail. That's an interface to the hail system for genetics and GWAS. And so we'll work with those. We'll explore Basilisk as user and as developer. It's important for developers who want their R code to make use of Python. It's also important for users who want to use those packages, because it does have some implications for what your infrastructure is going to look like, how long you're going to sit around waiting for a package to install and actually get running, and so forth. And then we'll look at some basic aspects of an analysis of CITE-seq data with scvi-tools. And we'll check a vignette that compares the scvi-tools results to the ASCA analysis. And then we'll restart R and explore the UK Biobank phenotype data with BioChail. That's the roadmap. Let's see how we're doing with our little workshop. Still just launching. And if you look at it here, it was created 4 minutes ago and has stopped. Stopped means it's still waiting to start. Still waiting to start. I would think so. I'm just going to be patient. We're going to talk for a while. We're going to just talk. Stop being started. All right. Thank you. Yours running? Yeah. You may have it come up here. All right. So wonderful books that everybody should have in on their disks, because it's free PDFs, Introduction to Statistical Learning. And one of them is in R, and very recently, they wrote one with applications in Python. So you have no excuse for not knowing how to do data science in Python. You now have this book. And I haven't spent as much time as I would like with this particular book. And I did spend a little bit of time thinking about what it would take to feel natural in the Python space. And already, we're on page 54 here, I'm starting to see these combinations of brackets and parentheses. And here's the array printed out here, an array of 0, 1, and so on. And there are so many different wonderful approaches to dealing with arrays in Python, like xarray is really important. And there's more to learn, right? But one of the things I take away from here is that there's probably work to be done to improve the interface. So we could write some modules, we could write some Python packages or whatever that would simplify and make it look a lot more like, it'd be just simpler. Here is how the linear models piece gets going, with a bunch of package declarations just to do linear models and regularization models. So again, this is one of the things we don't have to do with R. We get a library, something, and then you get an awful lot of infrastructure. But here, there's really a lot of packages you need to know about, and you import them with certain ways. It's fine. It's great. We have to learn it. And this is just something to keep in mind, that there are many people who take this for granted as the way to do their data science. And it's important to be able to reach to them and make sure that the stuff that we have done is used by them, usable by them. And there are reasons. There are reasons from a performance point of view, from a functionality point of view, that people are very focused on that ecosystem. And we do not want to fall by the side. We want to be able to take advantage of those things that they are so enthused about. And that's one of the reasons I feel very comfortable just saying, I want to see how scvi-tools works. I want to make it easy for people to use it if they just want to get involved with R and don't want to have to learn a lot of other stuff. And that is supported, that is aided by Reticulate, right, which is the Python to R interface set up by Posit. And one of the issues that Basilisk takes care of is the versioning, the pinning of the versions. A very important thing to be able to do. And we in the R world have a certain view of that. You declare the dependencies for your packages in your description file. You don't necessarily specify the versions, but the fact that you don't can actually lead to problems down the road. But it is still not part of the mindset of the R community that we should pin precise versions. And I don't have, I don't think we need to make a big change at this point to do that. But there are reasons that we don't have to. It has to do with the availability of CRAN as a very regulated distribution system. And I'm not going to get into that anymore. But the fact is that one of the great things about Basilisk is it lets you specify very clearly and have a very clear deployment of all the Python essentials for what it is you want to do with R and Python. And so what we have underlying our workshop, let's see how we're doing with that. Yeah.
So I don't know why you adopted it.
Yep. Maybe I should stop this one and try again, or should I just be patient? Just in the queue?
I launched one after.
Yeah, I believe you.
Anyway,
Yeah.
but yours is still not.
Yeah. Okay. How about should I kill it and try again?
No, I don't know if that will help.
Okay. Leave it. Don't worry, folks. It's only me. Everybody else has got a good workspace. I could steal somebody's URL. That's exactly what we don't want to do. But thank you, Andrew. It's a good idea. I could steal Alex's. That I can do. Okay, so this is what we're talking about here. This is the BioCPy interop workshop. And I've tried to spell out many of the things that we're trying to do in this workshop. And it would be helpful to have an acquaintance with a chapter of the OSCA book. Let's see what that looks like. This is the OSCA book, Integrating with Protein Abundance. So this is an analysis of a CITE-seq experiment. And it's a 10x PBMC, 10,000 cells. Doesn't really turn out to be 10,000 cells, 7,800 here. But this is what we do in order to get the data together for you. If you're using the OSCA book. By the way, interrupt me at any time if there's a question. It was interesting to see how the ScanPy folks go about the same type of operation. While I think what GetTestile will do is place this entity after some processing in a BioC file cache, my understanding of what the scvirs folks do is they will simply drop the resource down on whatever is the current folder that you're working in. So whereas there's a caching discipline here, it wasn't clear that there is a caching discipline when you work with the scvirs. Okay, no big deal. And then we go through the use of these libraries and structures in order to get to a place where we can think about the total abundance of the various cell surface markers that are measured with these so-called antibody-derived tags. So that's the CITE part of the CITE-seq. And then there's the mRNA. And what I wanted to do was go through the scvirs approach to getting to a place like this. Yeah, here. So we have the antibody-derived tag clusters, and we want to see how they lay out, and we want to be able to compare these to those. These are done in Bioconductor. We want to see how that compares to what happened in the scvirs world. And this display I thought was very interesting. This is saying, how can I look at signatures of the different clusters in terms of the cell surface markers from the CITE-seq experiment? Can we get here using what was done with scvi-tools, which is based on a variational autoencoder? So in a certain sense, there's a long sequence of steps leading to this analysis, this representation of the data, whereas with the scvirs, scvi-tools, it's more of a one-shot. I'm going to go and specify a model of a variational autoencoder And I'm going to get very close to this with what are called the denoised profile expression, profile abundance quantifications. So there's a lot with a completely different methodology. How similar are these? That was the objective of making this package. And then, of course, giving us access to the methodology. So you get a variational autoencoder, how do you train it, and so forth. You can actually try that out in this workshop. Okay, so that was this piece. We're looking at the, first we're looking at the OSCA book. Then there's the scvi vignette. Okay, so this is what I wrote in scviR. This is the CITE-seq example. And right away, we are able to run a function that goes and gets the anndata object that was made available through the scvi tutorial. And in particular, there's a variational autoencoder that you get by calling this function, provided you have the appropriate Python infrastructure running. And then you can see that their variational autoencoder has the is trained to be true. You can find out how it was actually fit. And you can look at the history of the training and on and on and on. Finally, one of the things I thought was very interesting was to look at the model itself. So this is what's called the module, which defines the autoencoder. So you can see all of the different tuning parameters that are set as we define the neural net elements leading to the variational autoencoder. This is something I have no experience with whatsoever, and I thought this would be a nice learning experience to go through and see, you know, is the inference based on this thing sensitive to some of these settings? So exposing all of this stuff is one of the reasons to do the scviR package.
If you want to go to that URL, there's an instance with your workshop planning.
Okay, so thank you. I'm going to just type in a new URL, https tinyurl.com.
Nobody do this, please.
Okay, Vince Manual, thank you. Okay.
And just delete
Now I'm going to
that.
delete that so nobody can do this, all right? Don't do it. You too, those of you who are out in the cloud. Okay, so let's just keep going through the slides, take it nice and easy. Yeah. Okay, so what we did here is we looked at the CITE-seq vignette. So this is part of the scviR package. You can do this anytime on your own. Just to go back, I think, to the BiocHail vignette. So I'm just trying to give you the roadmap of the overall workshop. So the BiocHail vignettes look like this. And what we're trying to do here is to understand what it means to use Hail, which has Spark behind, it has Spark, which is Java dependent, behind Python, which is giving you PySpark. And it was very nice to me to see that we can actually witness what goes on as you use Spark in this way. And in the workshop, you only have 2 cores for Spark to distribute its work to, but it does that. Does it completely autonomously. And if you had many cores and you had a cluster or whatever, it wouldn't take a lot of configuration to get a lot more throughput with this particular software. So that, to me, was very nice. So we will look at and run some of this code to get acquainted with Hail and the 1,000 genomes data. And I would really hope that we can work on some of the phenotype data that's just available for the UK Biobank through this. Also, BioC T2T. This was another thing which I thought was interesting. The T2T means the Telomere-to-Telomere build, reference build, very recently released, also computed at Johns Hopkins with a big group of people, but Mike Schatz was a major figure in the computation underlying this. And so with BioC T2T, you can see that here's the T2T, chromosome 1, Whereas if you look at chromosome 1 on HG38, there's 18 million positions that are not called. So there's a lot of additional information in the T2T genome. And one of the ways I thought it would be useful to make it available to people was by making a hail matrix for the 3,200,000 genomes. And I just worked on chromosome 17, and to make the hail matrix for this thing, it turns out to be, I think, 50 gigabytes zipped. But once you pull this down and you set some environment variables correctly, again, if you have a big machine, you can start running through this and looking, for example, at population stratification very easily with a new genome. So these are some of the motivations. I really want people to be able to move forward. I don't think we can do it with just R and Bioconductor the way it is. Things have been done in genetics and in deep learning that we don't want to reinvent. We want to just take advantage of the stuff that's there, learn about it, and if it's good enough, just use it. And that's why we have this little workshop. Any questions or comments before we start plunging in? Right. So, yeah.
Instance, I'm still stuck at stopped.
You're still stopped? I'm sorry, I can't help you. I did get one. How many people have running instances for this workshop? I know one. Whoa, I mean, okay. Thank you. Anybody else have it? Yeah. This is a 17 gigabyte image. Yeah. Okay, do we think that people will be able to get it, or?
Eventually, maybe. I just restarted all the servers, so I
Okay.
don't think it's cached anymore.
Ah, yes. So we had done some work previously. There was a crash of the academic cloud just this afternoon, and Alex noticed it, and he was able to get it rebuilt. Okay, so the hands-on part may be a little challenging to really work with. I'm going to see what I can think of. I actually, when I thought it was down, I started working with the POSIT cloud to see whether I could get enough stuff going so people could just make their own POSIT cloud accounts and get going. It doesn't seem all that straightforward to get exactly where we want to go. So you were able to get it running. I think the point is just to be patient. Yes?
There be a way to just download the vignette and run this template on the machines?
Yes. I'll tell you, we'll work through it a little bit, and I'll tell you what's problematic about it. Have you ever used Basilisk before?
Yes.
Okay, you've used Basilisk, so you know what goes on with Basilisk is about 2 gigabytes of Python will be downloaded before you can actually get going. And that's one of the exercises in the vignette. So here I am. If I do, so let's just see what it is. Okay, so I'll just show you a little bit about what's going on here. We'll start with the scviR. Sorry?
Each working on the ARM CPUs, the new Apple Silicon CPUs, you know?
Yes, it should work on ARM. It's Conda. Conda is.
No, I'm saying, but specifically about the BiocHail, about
Let me think for a minute.
the BiocHail.
There are some gaps for the M1, for the ARM stuff.
The BioConda package they don't build for them.
Yeah, there are still some gaps. I think the Hail one does work, but I don't think the scviR one works because it's a TensorFlow, and I don't think that's been all ported yet. Yeah. So you see, run anywhere is not quite working. Okay. So let me just show you how it would work. scvi equals scviR. And at this point, Basilisk is going out and assembling everything that's needed for me to get a reference to the scvi module. here, and you can see that one of the things that happens by default is there's a global seed for Python-based random number generation that has been set to 0. And now this SCVI is a reticulate module. There are issues here with how I'm doing this. And if you say names of SCVI, you'll see that there's a bunch of things that you can actually get access to here. And it's these that we take advantage of when we are working with the scviR package, okay? And one of the first things I wanted to do was see where these, to look at basilisk utils. And get external dir. Right. So this is a really important fact about what's going on here. Okay, so basilisk goes from version to version. And one version of basilisk may use a certain version of Miniconda, and another one uses a different version of Miniconda. And in order to give you good backwards compatibility with whatever it is you've done, every time basilisk gets a new version, it makes a new folder under the cache. And if we did here, I'm sure there are better ways to do this, but I'm going to do it this way. System, paste, du minus sh. Okay, I want to count how much disk space is being used there. Okay, it is 8.7 gigs, just for the software to do what we are doing here. Okay, thank you. Delete the URL. All right. So you can see, So we have had long discussions about, should we use the academic cloud? Should we use commercial cloud? And we're starting to learn some of the pain points of trying to rely on what I consider to be a really nice cloud, the NSF Jetstream 2. Yeah.
That was dying with the XSeed download?
The what download? XSeed download?
Yeah. For downloading the total VI processed 5K, 10K data, it was timing out.
Well, that's supposed to be coming from Open Storage Network, which is another NSF access project. And we've had no problems with that. But yeah, that's exactly what we're using. Let's see if that's going to cause problems for me here. So VAE equals get CITE-seq to VAE. This is how we get a copy of the, see, in my instance, I prepared a number of things so that the thing was already downloaded. I think that would not be the case for others. I'm not really sure how that could have happened here, to be honest with you. But this is going to, for you, it would actually download, I think it's not that large, all of the parameters of the fitted variational autoencoder.
That's not the part that's failing, it's the get total biz 5K, 10K data.
Yeah, the 5K, 10K, that's an anndata structure. Yeah, okay, well, we're learning. All right, well, still hasn't given me the variational autoencoder yet, so we're getting some real performance problems here. This is not, what am I trying to say here? You may go home saying, I don't ever want to do that. But the question is, what is the that? And here we are giving everybody in this room, in principle, would have a RStudio session with a good chunk of RAM and lots of disk. And I think what we're seeing is we didn't really anticipate just how much there would be. and whether there would be problems. So you really need, as you're going to offer something like this, you need a way of testing out the loads, the experiences, and so forth. And unfortunately, you're going to be a data point in this process here. Ideally, I would have used a different platform. It would have been easy to do this stuff on the NHGRI Anvil, and I think we would have had a lot more scalability there, probably. But you'd all have to have Google IDs and Anvil IDs and so forth. Yes, question?
Well, I wanted to learn more about the compute architecture you're using to deploy a lot of this documentation for the conference. Is there some website I could go to, or is there documentation for BioC about the architecture that you're using?
Alex will have a good answer to that.
Not to add more work, I'm just curious if it exists, and I could go and check it out. If I was interested.
This was a pet project that I started about 4 months ago, and right now it is not documented anywhere except in my head.
Well, I think that's a little. Let's try to unpack this a little bit. Okay. So first of all, what this is running on can be seen using the exosphere for the access NSF, right? So I usually have yeah. So you can learn about this through this document on exosphere, jetstreamcloud.org and so forth. Not exactly the right place to go for this, and my cache seems to have been lost. So the NSF Academic Cloud, Jetstream, yeah, I just want to show them.
I believe that's jetstream2.exosphere.app.
Yeah, okay. So I don't know where all my identity has gone. Yeah. That'll make me sign in. Okay. So I'm happy to do this because I think it's a very nice system. But if this stuff isn't cached, I'm not going to get there. And that's because I logged out of Google at one point. So learning about the Jetstream 2 and Access Exceed Academic Cloud is the first place to go, because that's the architecture. There's a bunch of VMs there. There's Kubernetes, et cetera. Okay. Now, as for what Alex did in order to have this nice Galaxy system, you say it's in your head, but clearly there's a code base. I don't know whether it's in a GitHub repo that's public or it's a private repo. It's in a Bitwarden, yeah. Right, there are certain secrets involved. But in principle, most of the code underlying this could be made open and documented, okay? There is another system that this was sort of inherited from called Orchestra, and that is, yeah, again, if my cache isn't here, I'm not able to do what I usually do. So the Orchestra Cancer Data Science. app.orchestra.cancerdatasci.org, I think is what it's called. I don't know if that's it or not, but the whole workshop system that Bioconductor worked on was created by Sean Davis, and that's what we were sort of, Alex has taken that and made it more maintainable by putting it under a Galaxy framework. So enough digression then about that. Any other questions about this? Because we're going to get back to the code and yeah, really sorry about this. But here we go. We got our variational autoencoder back. So that's VAE here. And we can say VAE, you can say names of VAE to find out what Python methods are available for this. And the one that I showed was VAE is trained, right? And it's true. And then if you were to say VAE train, it'll do 2 more iterations. And in this case, if there was a GPU behind this, it would use it, which is kind of nice. There isn't one here, so it's going to have to, throw the data into the CPU and try to update the parameters of the variational autoencoder. And this, it will work. And this is a way of starting to get a handle on the sensitivity. I guess we lost it here. We'll get it in a minute. I'm going to just kill that.
The
Yeah.
VAE object, that includes the single cell data that it was trained on originally as well? It's not just the model?
Yeah, I think that's right. That there's a lot of stuff in there related to the anndata that was used to train it.
Right.
Yeah. Yeah. You see how it says here the model's A data is minified, false? That's because more recently they have some compression or data reduction methodology into the in the representation of the VAE that was not used for this particular instance. So let's see if I still have this object here. Yes, I do. Okay, good. So we're bouncing back and forth. I guess I'm using too many resources. But remember, scvi module, or no, it's VAE module, right? VAE module gives you the code of the autoencoder, and it was precisely these sorts of tuning parameters that I wanted to start to explore to see just how sensitive the inferences might be. Okay, so that's the variational autoencoder. Let's take a look back at the vignette. Right. So what we wanted to do was work through some of this. So you're saying, Tim, that when you try to run this piece, you were crashing.
Well, I found out why. The download speed is incredibly slow, so I'm stepping backwards.
Yeah. The download speed. I mean, if you're doing it on your laptop though, right? See, the hope that I had was that since we're running in the cloud and we're talking to OSN, all of the students here would be able to get pretty fast downloads of these things. So let's see whether my system does it. Right. So here we are. Here we go. There you are. Now, it could be that it was cached. So, yeah, in my particular image, I don't know why, if I got it from Hedia, that it would be cached, but you see that this happened pretty quickly. It's my Docker image. Okay, so I did pre-compute a number of these things. I don't know whether I did that one. Let's remove it. Yeah. Let's look at what this thing is. Okay, this is a function. You should be able to look at the source code and understand what's happening. Yeah, cache CITE-seq 510 PBCMS. So that's the function we need to look at. Run it in the debugger. So H5 path here. Yeah. So that's what you need to get. So if we look at this piece of code, you'll see that what it does is it tries to get out of the OSN an H5AD, which is the CITE-seq 5K10K entity, and it puts it in cache if it's not there.
Since it's using download file, like actually the default timeout in R is really slow. You can change the option timeout.
If you're doing it on your laptop.
Yeah.
Yeah.
If you don't have any timeout problem.
Right. Yes. Yeah. Okay.
The default timeout is like 1 minute or something.
Okay, so how are we doing? We're 45 minutes into the workshop. I think it's a good idea to take a break. And then we'll come back and we'll wrap up the scvi stuff, and then we'll talk about hail, and then we'll knock off, okay? So let's come back in about 10 minutes.
I mean, I've seen you on
Nobody has made a decision. I would say, I mean, the tradition was CITE-seq for pricing stuff.
My team, we all come down. We have to use one day. I come in the office.
I think it's possible. I think you should ask not a lot of have practice here.
Yeah.
As long as Python can get
Well, they could do self-training. Yeah.
so now this is the vignette for this workshop. And as long as they can get it, and we can just get the
Yeah.
they can hit it and they can go step by step. However you want to do it.
Yeah.
So that's one problem. Someone offers an error markdown, you make it available to the students. How do the dependencies get solved? We want to make sure everything is installed in advance.
Yeah, so I want to try to do it.
Those subproblems are solved. Getting to a point where you have a platform like this, so people can think through any of these workshops that they'd like to do. That's what Alex is doing.
Oh.
So that actually your author can just file a GitHub issue in a certain place, telling the app where their vignette is, and it goes and produces a document.
Okay, yeah.
I think it's reasonable.
Yeah.
Okay. And how to take advantage of other people for studio tasks. I don't have much to say about that. This particular format is pretty effective.
Right.
This is the first time that I think it's ever been.
Well, it's probably all everybody at the same time.
Yeah.
So I've experienced this kind of training before. When I have a Shiny app. People are demoing their Shiny app. I got to put it on a physical server with a bunch of core and a bunch of memory. Otherwise, it just falls over. So, all right, this is great, by the way. I like the way it's set up. But if it would go, I knew it would be. I was looking at the way it's structured. It looks like it really well done.
We're happy to come over if you want to.
Okay. Yeah, because I'm right in the either Cambridge site or we're in Longwood. And go along.
I park my bike right next to it.
Oh, okay, yeah. All right. Okay. Okay. All right. Appreciate it.
I don't know why, but Marcel's is launching, Glow is launching. I don't know if it's because it's just a big one or has hail, or but it's just this Docker image that isn't. Or yeah, I have no idea why.
Well, we need to have a platform where we can scale it. So.
But it's not capacity. You can launch any other workshop and it's launching within less than a minute. It's just this Docker.
Yeah, okay. So we'll just have to look at it ourselves and see. Can you do some profiling? Or?
I mean, I don't know.
Right.
I think, one thing we could do is stop using custom Docker images. So have them all be built by the way I'm building them for the package demos, for example, and not have people have a separate Docker file. Because, I don't know what in this image is making it special, but it is something related with this document. It's just pulling from your GitHub container registry. So, I don't know.
Well, it's just a topic that would be
we
good for
should.
us to dig into and try.
Yeah.
How do you simulate something like this? I don't know. But that's what we want to be able to do. Okay, folks, we've had our 10 minutes. Let's try to get back on track. So apropos the underlying architecture, I did find the Jetstream 2 PDF. We'll put that in the Slack eventually, but there is a paper about it. And, you know, in every other circumstance, I think we have found it to be quite reliable. And the issues with the this particular workshop, which has a 17 gigabyte Docker image, that I'm responsible for the problem there. Maybe if I had split it into two, then we would not be experiencing this. But I wanted to try and do both the single cell and the hail. I think we'll transition after wrapping up a little bit with the single cell. Let me just take us through a little more of the vignette, and you can see what can be done here. So we get the anndata object. We have a function that gets the autoencoder, and you know, it's nice to see. Here's one of the features of this business of deep learning that you know, again, I'm not really well versed in it at all. But what you can see here, this negative ELBO, it's like a goodness of fit criterion, and in a Bayesian variational framework. And it does not change much in the validation step after even 150 epochs of training. And the question is whether you know, we are eating up too many CPU cycles and waiting too long without, you know, doing much to improve the fit of the autoencoder to the data. And the pre-specification of the number of epochs is, I don't know if anybody's doing a lot of deep learning here. It seems to be typical. And there is some literature indicating that people are tending to use more epochs than they need to. And understanding, you know, when to stop is, you know, I think it's not a solved problem. And there are local minima and so forth that you don't want to get trapped in. So I really don't know what the guidelines are for this, but one can see pretty clearly that this may have been run too long. What I wanted to know is if I run it a few more iterations, do things change very much? And so far, what I see is that actually some aspects of it do. And so that's where I would like to spend some more time trying to understand this. Then the number of computations we need to do in order to get to the clustering is really very small. And then once you have this, the code is available in the vignettes to compare identities of clusters found in this procedure versus those found in the OSCA book. The adequacy of the batch correction here is presented just the way they do it in the vignette, in the tutorial at scvi. And finally, to see how the different clusters vary in terms of the locations of cells that have certain protein markers is easily computed here. So I'm going to stop there. This code is available. You can run it on your laptop, but you'll have to actually get the scvi package installed, when you run scviR the first time, Basilisk will go and find all of your pip dependencies. It will use a miniconda environment to place them in the cache, and they will all be usable for you henceforth until either the version of scviR changes or the version of Basilisk changes, in which case that will be done again to have something that's coordinated with the current releases. Okay? So let's try it out with hail. Let's see if we have better results. So I'm going to stop and restart because the Python infrastructure can't be shared between hail and scvi-tools. So let's give it a try. Library BiocHail.
Delete the link.
Delete the link, yes. Okay. And we will say example, get 1 kg. So there is, oh, I must have done it wrong. We're going to do an example where we are using some 1,000 genomes data that are made for a tutorial by the Hail folks. And so what's happening here is hail init is being run. That is a Python method on the hail module, and it will start Spark. There we are. So now we are running this example. We have a get 1 kg on the HL, which is the hail environment, and we have a matrix table. This is the great creation of the hail folks, that they use the matrix tables in the Spark framework to represent vast quantities of genetic data. And so we're able to work with these things pretty conveniently with this package. We can start learning about it. And in particular, if we're very ambitious and we want to get 50 gigabytes, we can get 3,000 calls of all of chromosome 17 on 3,202 people in the T2T reference genome. So all of that stuff is covered in the packages that I described to you. What we will work on right now is the first vignette in the BiocHail collection. Google on that. We got this. Here you see that among 2,200 packages in Bioconductor, BiocHail is down at 2226. So it is not widely used. And I don't know whether that reflects an aspect of its quality, but why don't you just check it out and maybe I'll get a better rank. So we can see the different things that are being done. Acquire a slice of the 1,000 genomes, genotypes, and annotations. So there we go. BiocHail can be installed, and we'll get ggplot2 also. We run hail init, as I showed you. It takes care of getting the PySpark and the Spark in place, and we can work with matrix tables. So the first thing you want to know is you have an object called a matrix table, right? MT, and it's an instance. And this reference here is the way Reticulate will expose the matrix table to you. You can say names on it and find out all the different methods that can be used. So one is MT describe, tells me a lot about what's in there. And you can start to see some relationships between what's in this matrix table and what you would expect to see in a VCF. And in fact, this is a transformation from a VCF for the 1,000 genomes. And there are other methods to actually work with the genotypes rather than just using what we remember from VCF tools or whatever it is we like to work with. I think there's a shape also. No, so I don't remember how to get the dimensions of it, but maybe it's in the vignette. So there are rows methods and select methods and show methods, and this is how we can show some of the rows of the matrix table here, the locus and the alleles. And this is done using Quarto, actually, so you can see that I can make references to the Python object, prefacing them by R here. We use the prefix R to find references defined in our R session. Okay. And then MT count is what tells me what the dimensions of this matrix table is. And we can rewrite now, we can write a dim method for it if we know what we want to use. And so this is writing a dim method for matrix tables, and you can see that it gives back what we expect, the number of rows and the number of columns in a vector. Okay, so we can do some programming to make matrix tables behave a little more like R matrices. Then we have the describe. And here we can use the R to Python capabilities of Quarto to add sample annotation to the matrix table. And ultimately, we're going to actually work with the variance, do some quality assessment, get a histogram of read depths, and see the call rates, filter for sample quality, and finally run a GWAS with a fabricated phenotype. So if you wanted to do this on your own in RStudio, what would the best way be? Here's what I'm going to propose. We start a new project. It's going to be a version control project, and we're going to clone from Bioconductor's Git. Packages, I see hail. So this you could do on your own RStudio, create the project. It pulls down the source image of the package, puts it in your home, and now I can go to BiocHail folder, vignettes, and I can get the GWAS tutorial vignette into scope here. And I can run that one chunk at a time. So that's Control-Alt-N. I'm not doing any package updates, whoops. Control-Alt-N, at our libraries. We can run hail init repeatedly because we set a parameter called idempotent equal to true. And we can go through all the steps of the vignette.
That's just a
Yes.
general question about interface between R and Python. Is there any computational overhead of controlling Python via R? And if so, does it vary by process or package, and how much? Benchmarking on that?
Right. I think the basic view of that is that there is not a lot of overhead, that one is working directly with the references to the Python environment there, thanks to the way Reticulate was implemented. You might find some overhead introduced in the Basilisk framework, which you can control, because it will actually start a separate process through a function called basilisk run. Actually, you know, I haven't paid as much attention to the workshop as I should have. There's so many issues here. But let me take you through that just for a minute. Sure. Yeah, there was a piece of this for developers that I wanted to make sure we looked at. So if we look at BioC Pi interop vignette, wherever that is, home. Yes, this one. Okay. So let's blow this up a little bit more. Yeah, actually just knit it, right? So that should work. That should work. Okay, so we're knitted it. We're going to try again. This is what I wanted to talk through as at the beginning of the workshop. And what we're talking about here are the details of coding an interface. I'm going to try and blow this thing up. You can see it better. Yeah. So when I wrote the scvi-R package, part of it has a basilisk.r in the R folder, and it sets up the environment by giving it a name, so other functions can refer to it, the package that it's in, and then we're pinning the versions of various Python packages that we know we need. And sometimes there may be a gap here, and I think Stefano may have found some problem with JAX. I've run into problems with that as well. But anyway, this is the way you define the Python environment that you want to have fixed for your use of this R package. And then when it's time to actually write a function that uses the Python, this is a very trivial one. It starts basilisk, runs basilisk run here with a process that will use that environment. And it runs reticulate import just to bring in the module scvi. Now, in general, you'll want to use, in your function, you'll want to have a protected approach in a separated process to using the Python. But in this case, I'm just handing back this reference. This may not be the safest way to do this, but it seems to work okay in this context, okay? Usually we just use the Python stuff within our function and we hand back what we want to hand back. So I wanted to make sure folks are clear on how that is being done. Then when you actually are, the first time you use your package, you install it, it installs fine, but you call the function that is going to use Basilisk, it will run a Miniconda installer, and that actually usually takes up a fair amount of bandwidth and a certain amount of time to produce what I showed you in the cache before, which is the Basilisk version that has the necessary Python infrastructure for every package that has declared itself as a Basilisk client, in essence, okay? So the way in which these processes is handled can lead to some overhead. But I think the way it's been done allows you to do parallel computing with the Basilisk environment and so forth in a safe way.
One reason that I actually really like Basilisk over Reticulate is the ability to better switch between environments. You have a Conda environment that you use for one set of processes and another one used for another set. With Reticulate, you usually have to reset the entire R session. And with Basilisk, I find it's easier to switch back and forth. And I've written some extensions to even take that a step further and just find the paths to the executables within a Conda environment and create a system wrapper for that. Probably not the safest way to do it, but yeah, it's another attempt to make that easier switching. But I think that is something Basilisk does better.
Yeah, so for those online, if you didn't hear that, Brian Schilder has some remarks on how to use Basilisk to take advantage of multiple Python environments at once. And hopefully some documentation will come forward on that particular technique. We'd be happy to see that in Bioconductor. Okay, so what we've been through here is some of the material for developers as to how you would use Basilisk in your own package code if you're going to use Python. And here is the example where I showed you 8 gigabytes was being used by the Basilisk infrastructure. If you actually look at the amount of code of disk space used by R, it's less than a gigabyte. So it's worth knowing. Then we use these commands to run scvi. We did that. And a very nice thing about Reticulate is that you can get Python help easily. I'll show you how that works here. So let's say I want to do py help HL in this case. Yeah, I have to say library. I have to bring in Reticulate to begin with. Yeah, py help. So HL was the name of my Hail reference, and I get information about the different components of it. And I can go further by doing, for example, $FS, any of the modules there. Yeah, well, that would be cool, right? How do they do Fisher's exact test in hail? Well, you can get help on that and compare it to what R does. All right, so that's a little bit more on this, the background here. And then we ran some of the vignette code, right? I jumped around a little bit. If I go back to my vignette here, which is, get rid of that, get rid of that. We're back in the GWAS tutorial. Let's just knit this and see if it will go. I think I want to do it like this. Yeah, let's just knit. Aha, so it fails. And I think it fails because there's no BioC style. So I forgot to include that. Let's take care of that. Okay. That was easy enough to solve. We won't do any more. And let's try and knit it again. Okay. So my point in doing this is to indicate to you that if you use this workshop system on your own without many people running it all at the same time, you can actually work through these things for the next 6 days at least on your own and find out whether there are problems or interesting findings that you'd like to report to me. I have GitHub repos for all of this stuff. You can file issues and let me know whether this is helpful in thinking about R and Python interoperation. Here's an interesting part. This is where we're using Spark. We only have 2 cores, and every time Spark is starting some parallel computing, you can see the stages that it's going through. In this case, just to do a QQ plot of all of the p-values that are available in this GWAS, you can see how it steps through. And if you had 20 cores, this could go up to 20 simultaneous jobs that it would take care of on its own. So the vignette ran nicely. It would have run on your place if we had done the right things in terms of capacity. This was just computed for you, and it includes all the computations involving matrix investigation, looking at how the genotypes are actually represented and getting counts of them, histogram of read depths, and finally, a QQ plot for GWAS, which is then refined by estimating population stratification through PCA, readjusting, and then getting, we don't show you the new QQ plot, but that would be an exercise. We can see that the lambda for genomic control goes down to 1.08, and then we get a Manhattan plot for the simulated data that shows that the phenotype that we were interested in can be detected with the genotypes. So that vignette runs there. Let's see if there's another one in the hail world that might be of interest. So again, remember, because I made the Git repo, not much of an R studio person. Here we are. BioC hail in our vignettes. We have the large T to T. So this is describing how you would pull down chromosome 17 genotypes for the 1,000 genomes folks. This is telling you how the matrix, the matrix table was made. Why did I do this? Because I wanted to show that we can do scalable computing more readily with the matrix table from hail than we can with a genotype VCF that is being distributed at the 1,000 genomes. So it was very easy to make this conversion, although it took some time. Here it was. Hail import VCF, this large GCF file, write it out as a T to T 22.mt. This one is chromosome 22. I also did it for chromosome 17, and that's living in the open storage network where I describe here. And once you have that, you can do the population stratification study that is shown in the compiled vignette. And so there are some exercises here. You know, I do the population stratification exercise with a 1% sample. You can also do it with a 5% sample with very simple code. Is there gain in information? Find the geographic origins of donors of the 1,000 genomes, genotypes, bind them to the MT17, and then color the PCA plot. Produce an artificial phenotype and find which loci are most strongly associated with that, and then make an artificial phenotype that is associated with geographic origin and show that by using the PCA, which is readily computable, using the code in the other vignette, that you can actually adjust this out and recover the appropriate false discovery rate. Any questions online? Yes.
Question online. Can basilisk deal with different versions of GCC? Say Python environment is based on GCC9, but R is based on GCC13. I don't know the answer.
Different versions of GCC, I think, are a no-no.
Okay.
So your run times are going to be incompatible with one another.
In principle, basilisk supports a separate process configuration, I think.
Yes.
Where you can communicate. I don't know how efficient it is,
Uh-huh.
but if you were to do that, it should work.
Okay.
I also encountered this problem with Bioconductor, and I basically wrote some system wrappers to find the right GCC version for whatever you're running, and then it loads some environmental variables and sets that, and it seems to work.
Okay, I stand corrected. Yeah, Aaron actually did some extra work looking for glibc incompatibilities. And there's something called preload or something like that where you can test to see whether there is an incompatibility. So good question. There's more scope to doing it than I thought. And if you have problems, just file issues and we'll see whether we can get to the bottom of it. Anything else? I mean, for an online question, that's a doozy. That's not what this was intended for, but I'm glad we could address it. Well, you've all been very patient and tolerant of a surprising situation, but I hope you learned something from this. I'm ready to wrap up. But not because I don't want to answer any more questions or do any more exercises. If you have some questions, please let me know. But I think this is yeah. Did you have a question, Andrew?
Yeah, how about data compatibility from R to basilisk? Like I, ran some, liked how I did some analysis with like a single cell experiment, but now I want to run scvi on it.
Right. You're familiar with Zell converter?
I've heard of it.
Okay.
I haven't used it in Anchor ever.
Uh-huh. Yeah. So I think there are many approaches that might work here. But Zell converter does have an SCE to anndata converter in it. And I think it is pretty good in terms of not losing much information and presenting it to anndata in the way that is appropriate. So that would be one way to go forward. I think there's enough work in the scvi space with anndata and R that, you could see what goes wrong. And again, I think we can work around it, but I don't think there's any major obstacles. I mean, there is HDF5 behind the anndata structure, but there's HDF5 behind ours also. We can handle different flavors of matrix organization in HDF5. You would probably know more about that than I do. And so I think there are no roadblocks in terms of just the data representation. Yes, Adine.
So when you're thinking of BioCPI, like C version of the classes, how will this, how does it all work together, and how will it all the advantages?
We have Jayaram here, who is the BioCPI author. We had a discussion of this over lunch. I think from a strategic point of view, how we're going to present this to a larger community is a little bit up in the air. But if Jairam wants to make any comments about the roadmap for BioCPI or whatever, certainly hand over the floor.
Yeah, I mean, the current implementations or at least the current goal is to bring BioC representations to Python. And once we have, a pretty good set of those classes implemented, we can start thinking of what the next set of steps would be. But going back to one of your earlier questions, if you don't want to go the self-converter route, there's a package in BioCPI called RDS-Py that can directly read RDS files in Python and give you a single cell experiment object that you can then use any of the other packages in Python.
Yeah, so for those who are interested, there's a lot that's been done by Aaron and Jairam at Genentech, others undoubtedly, on this notion of language agnostic representations of data for genomics. And the packages to look at are Alabaster, which holds a lot of the transformation from RDS, let's say, or RDA to a more language independent format, maybe HDF5 for the numerical data and JSON for the metadata and CSV for those things that are representable as CSV. So you're trying to get away from the requirement that you're using a specific scripting language to work with the data. Once you've got it in this language agnostic format, you can work with it with many other languages. But the definition of client packages, client functions, let's say, for a given language that suck in this agnostic data and give you what you would like to see. In the case of Bioconductor, it might be a G-ranges or biostrings or something like that. Some work has been done in that space and more needs to be done.
I should also mention that at the EuroBioC conference this year, there is going to be a pre-workshop, basically a hackathon on anndata R, which is an R native implementation of anndata. And so, yeah, that'll be interesting to see how that works with the packages like this.
Yeah,
it will be recognized, but I think those are all things we'll want to keep in mind.
So that's distinct from, say, Martin's effort around mapping and data.
Exactly. Yeah, it's a different strategy.
You're now saying we want an anndata-like structure implemented within.
Exactly.
Even though we already have some as a spare.
Yes.
So
yes, exactly. Well, it would kind of be like, yeah, it would be the next generation anndata objects within R. In theory.
So for those online, there is a discussion of an anndata R hackathon that will take place at EuroBioC in Belgium later this year. All right. I think that wraps it up. Again, I thank you for your patience and forbearance. And we're going to have a poster session tonight, I think, today. Yes, so let's all get together for that. I don't know what's happening with lightning talks, but tomorrow. Okay, so please, if you haven't signed up for one, think about it. Thanks, everybody.